Everybody, thank you for joining us today for the Meet the Scientist. I'm Jaquel Quiroz, Associate Professor at the Harvard Medical School, and it's my pleasure to welcome you to the Meet the Scientist today, that we're going to be meeting the team of the Translational Neuroscience team at the Michigan State University. So as a reminder, the Meet the Scientist webinar series is an opportunity to meet investigators that are working to end Alzheimer's disease and other dementias, especially an opportunity for junior investigators to learn about the exciting research that these teams are doing, and also an opportunity to learn about career opportunities available to them. So as usual, please use the button for the QA to ask any questions. We are going to save some time at the end to go through all those questions. Please ask the questions as the presentations are going, and I'm going to be leading that discussion at the end. So with that, I'm going to pass it to Dr. Irving Vega. He's an associate professor at the Michigan State University. Thank you so much to all the speakers for being here and everybody watching as well. Thank you. Dr. Vega.
Thank you, Jaquel. Gonna start sharing the screen now. Okay. Okay. So again, Jaquel, thank you very much for inviting us to this great opportunity to talk to junior investigators. This is the team here at Translational Neuroscience that we're going to talk to you in different aspects that we work on Alzheimer's disease and related dementias. I'm going to start, and then it's going to follow by Nick Kanaan, then Marcia Gordon, Scott Counts, and then Dave Morgan. And we're going to continue the discussion about the work that we do on Alzheimer's disease. We are based in Grand Rapids, even though it's Michigan State is associated with Lansing. The headquarters of Michigan's, the College of Human Medicine, the medical school is here in Grand Rapids in this building here that is called the Sequia Center. And I'm talking to you from actually this office right here. And then to the side is Scott, and then Nick with offices around here. Then Marcia and Dave are around here in the 4th floor. This is the main building. I had to update this photo because in Grand Rapids, the Michigan State University footprint continued to increase. We have a new building at this side right here called the Doug Myers Medical Innovation Building that is already up. And then back here, we have a new parking deck and also a new building that is going to be the North America headquarters of Perrigo. That is a pharmaceutical that develop healthcare products. And the headquarters are going to be here in North American headquarters are going to be here in Grand Rapids. And we expect to have collaborations with them as well. So as you see, the Michigan State University, especially the College of Medicine footprint, continue to increase here in Grand Rapids. And this is actually called the medical mile. There's hospitals and a lot of activities, research activity happening, including the Van Andel Research Institute. So my work, now transitioning to what we do individually, my work is related to the identification of biomarkers and novel modulators of tau-mediated neurodegeneration. So I'm interested in understanding the process that leads to the aggregation of tau and also the response to the toxic forms of tau. So the scientific premise that we have is that tau is a structure is very dynamic. And that dynamic goes from monomers to dimer, trimers, and then oligomers. And we believe that there's a lot of post-translational modification and protein-protein interactions that facilitates these transitions between monomer to small oligomers and perhaps going back. But at some point, tau gets stuck in toxic conformations that are no longer transitioned back to physiological forms. And once the protein gets stuck in those structures, the toxicity increases. But there's also another response that if the process of aggregation continue, it goes to tangles that tend to be, and there's evidence that suggests that they're less toxic than the oligomeric forms of tau. So our scientific premise is that there are protein-protein interactions and post-translational modifications that modulates those transitions of regulating the changes in the structure of tau to go from physiological forms to pathological forms. So in order to understand, identify those proteins and also to identify changes in the proteome associated with the accumulation of pathological tau, we use mass spec proteomics approaches, and we take from tissue, from human tissue to animal tissue, and also we are currently using also biofluids to identify changes in the proteome associated with the accumulation of pathological tau. We have identified several proteins, and we go from that discovery phase to then more of a hypothesis-based approach. So one example that I want to tell you today that we move from the discovery to the hypothesis-based approach is the identification of this protein that we call that was called EFN2, EFHD2, for EFN containing protein 2. And we found the association of EFN2 with tau in a tauopathy mouse model that expressed the human tau with the P301L mutation. And this protein is a calcium binding protein. And again, this was actually identified, we identified in the mouse model, but we went and looked at human tissue and Alzheimer's disease tissue. And we found that EFN2 colocalized in the somatodendric compartment with pathological forms of tau highlighted here by the phosphospecific antibody, tau antibody PHF1. And the colocalization is mainly in the somatodendric compartment, suggesting that EFN2 is associated with aggregated tau. So we started to study EFN2 and tau in vitro, and using liquid-liquid phase separation in collaboration with my colleague Nick Kanaan, we found that EFN2, when incubated with tau, actually transitioned tau from a very dynamic protein in solution to a solid phase structure that by FRAP assay, after photo bleaching, it doesn't recover. But in presence of calcium, both proteins become very dynamic, and they're together in those doublets. And then after photo bleaching, they can actually recover, meaning that EFN2 modulates tau dynamics in vitro, but apparently influenced by calcium, by the presence or absence of calcium. We did some in vitro, continued in vitro board characterization. As you probably know, in vitro human tau needs inducers to form filaments, and in this case we use arachidoic acid. And we found, like, we see, like many others, that arachidoic acid induce the formation of tau filaments and oligomers in solution. When we add EFN2 to the solution, what we found was like tangle-like structure that by immunogold, we know that tau and EFN2 are localized together. So now with this information, we cross the tauopathy mouse model that expressed the human P301L mutant tau with the knockout EFN2, and now we're trying to characterize in vivo what is the role of EFN2 in the progression of tau-mediated neurodegeneration. And in this animal, the phenotype is motor impairment, easy to measure because they have degeneration in the spinal cord. And by looking at the motor impairment, heterozygous and homozygous knockout early on developed that motor impairment phenotype. And we also see that in the knockout, transgenic knockout for EFN2, there is more accumulation of phosphospecific sites of tau that are associated more with pretangle formation, suggesting that maybe EFN2 promotes that transition from oligomeriform to perhaps tangles, and it might be a neuroprotective protein by sequestrating pathological tau in a tangle-like form. And we are undergoing these studies. So in summary, we go from a discovery-based approach to a hypothesis-based where we try to characterize the biomarkers that we identify. And this work has been done by many now currently in the lab and past members in the lab, my collaborators, and funding that support all this work. So with that, I'm gonna pass the now the turn to my colleague, Nick Kanaan.
Yeah, thanks, Irving. Good afternoon, everybody. Thanks for the invitation, Yakil, to come and share our work with you today. I'm Nick Kanaan. I'm an associate professor in translational neuroscience. I guess I've been working in aging and aging-related neurodegenerative diseases pretty much my entire career, all the way back to undergrad, where I did Huntington's disease, and I switched over to Parkinson's and aging in my PhD, and then moved into tau and Alzheimer's disease and other dementias in my postdoc, and have continued that for about 15 years now. And I focus very heavily on tau. So when people ask me what I'm into, I jokingly say anything and everything tau, as you can see in my title here. But more serious than that, there are several areas that we're very interested in and have been studying, some more recently and some for several years now. It includes trying to understand the molecular mechanisms of how tau can directly cause dysfunction and degeneration in the context of disease. We're also very interested in just the normal physiology of tau and where it is and what it's doing. On a more protein level, we're interested in connecting tau structure with tau function, or in the context of disease dysfunction. And then we have a relatively unique expertise in making novel monoclonal antibodies, which we've historically used to study specific forms of tau. And the antibodies would allow us to kind of connect the dots between our in vitro work, our cell and animal work, and human disease tissue. But more recently, we've been leveraging the antibodies we have for more clinical applications, which is where we get into novel disease-associated biomarkers. So these are the areas that we generally study. I mean, there's obviously a lot here, but I'll highlight a few of the more recent things we've put out there and what we're kind of actively pursuing currently. We've always been interested, again, in normal physiology, and one of the questions that I think is central to starting to pursue our better understanding of how tau could play a role in normal and disease physiology is understanding its distribution. And many of you may have heard through reading and such that tau is an axon-specific or an axon-enriched protein, and it's really not found in other places. And I'm here to tell you that that's absolutely false and has been for the last 40 years when it was initially described to be found in the nucleus, the soma, the dendrites, and the axons, as well as oligodendrocytes. And so in a recent paper, we took a host of antibodies with different epitopes through the protein and found, again, we confirmed this historical finding that tau is not axon specific, but it's everywhere, pretty much in neurons. And in a subset of glial cells, it's mature oligodendrocytes is the main source of glial tau in normal brains. But probably the more interesting point was that different antibodies with different epitopes highlighted different pools of tau, which to me indicates that it could be likely that tau is held in different states in different cellular compartments. So that's an area of active investigation for us that we're pursuing. And it's pretty exciting stuff, I think. So you can go to the next slide, Irving. Another area that we've generally been pursuing is related to the idea of tau function. Again, many of you may have heard through reading that tau is primarily involved in stabilizing microtubules. I would argue that that is very unlikely its main role, and certainly not its only role, if it is one at all. And this is supported by several lines of evidence that, again, like I just told you, place tau in different compartments, some of which don't have microtubules. And this host of studies that have really been trying to dissect tau's interaction with other proteins and the ways that it can regulate their function in various contexts and various compartments in a neuron. We focused very heavily on the axon, but really I would pitch that tau's main function is to act as a signaling hub or a scaffolding-like protein. And sorry, this is sort of busy, but tau is in a lot of places and it has a lot of different interacting partners, which is very consistent with an intrinsically disordered protein. Irving, you can go to the next slide. The last sort of basic science area I'll highlight is something we've been pursuing for many years now. You can see the list of references on the right over here. And it's trying to understand a specific mechanism by which we think pathological tau can cause axonal dysfunction and ultimately degeneration. And it connects structure and dysfunction and specific signaling pathways. And the gist of this is that we've been able to identify in several pathogenic forms of tau, but specific forms of tau, that the structure is changed in a way that exposes a motif in the N-terminus of the protein. And when that part is exposed, it can trigger the signaling pathway that involves protein phosphatase 1, GSK3, affecting kinesin, and then that causes cargo dissociation from the motor complex, effectively impairing axonal transport. And again, you can see the list of papers that we've been engaged in over the last several years. And I'll highlight one now that is very recent. I think it was maybe out last week or the week before, from a research assistant professor in the lab who's really talented, published his work looking at some FTDP17 mutant forms of tau and how they affect axonal transport. And it's a really nice paper that translates our work that we've done in squid to mammalian cells, identifies a specific, the specific role of PP1 gamma, actually. It's really nice work. And he also was lucky enough to get the cover shot. So kudos to Ben. You can go to the next one. And the last area I'll talk about is sort of this newer branch of our research program where we're leveraging our antibodies that we've made against these various special forms of tau and pursuing them in the context of biomarkers for AD and ADRDs. Coincidentally, several of us, actually all of us, are involved in the Michigan Alzheimer's Disease Research Center. I lead the biomarker core with my colleague, Dave Morgan, who you'll hear from shortly. Irving is also very directly involved with our mass spec work, but the three main goals are to measure existing blood biomarkers as you would predict. The biomarker core in the MADRC is new, so it was just added in this last round of funding. So we're getting geared up and moving along, but one of probably the most exciting goals, I think, is that we're going to spend a significant amount of effort trying to develop novel assays that will include using, again, some antibodies that we and other investigators in the group have with a Simoa platform, which is basically an ultrasensitive immunoassay, and mass spec to identify some new targets to pursue through Irving's mass spec unit. And of course, we'll integrate what we find into the clinical landscape and see if we can really make some advances there. So that's really exciting new avenues we're pursuing. You can go to the next one, Irving. Okay, before I get to my last slide, thanking everybody, I'll say that I have a postdoc position open. Actually, I think it's for a couple, but nonetheless, go look at the posting, reach out to me if you're interested. Please spread the word amongst your circles. I would love to add people to the team. So you can go to the next one, which is the team.
It's coming. Oh, what's happening? Come on. There we go.
Yeah, thank you. Yeah, these are the people that make the magic happen in the lab. Of course, I have all the people that are directly in my lab and my collaborators listed here. They obviously move the work along, and I'm really grateful for the work that they do. Of course, we have been very fortunate in getting funding to move things down the line. So that's always great. NIH, Health Association, Bright Focus, Standard, AD related funding agencies. So with that, I'll wrap up and hand the baton over to Marcia. Yes.
Okay, so I'm Marcia Gordon, and I'm a professor in translational neuroscience, and I am much more cell-based and system-based than protein-based in terms of my interests. And so I'm going to talk a little bit about innate immunity. So you all know from COVID that your immune system can generate antibodies that are very specific against pathogens. But you may not appreciate that there's also another aspect of the immune system that doesn't generate specific antibodies mediated by different kinds of cells. And they recognize general patterns that are found associated with infectious agents and also damage to tissue. And they respond much more generally. And so these are some photomicrographs I took many years ago. The pink dots in the larger pink areas are areas of amyloid plaques in transgenic mouse models of Alzheimer's disease. And surrounding those amyloid plaques are clusters of activated microglia and astrocytes. And that's the activation of innate immunity in the brain. And you might think that that is a response to the accumulation of abnormal proteins in the brain, but actually a lot of genetic evidence is now suggesting that there are many genes, as many as 30 or possibly more genes, that contribute to risk of developing Alzheimer's disease. And many of those genes are associated with innate immune activation. So there may be more causative factors associated with immune activation that contribute to Alzheimer pathogenesis. Next slide. So, we thought that innate immune activation would not necessarily be a good thing. And so we tried some experiments where we directly activated innate immunity. And in the panels on the left-hand side, you can see two different stains for microglial cells as a function of time after this activating stimulus. And you can see that after 1 day and 3 days and 7 days, more and more of the glial cells respond and become activated in response to the stimulus. However, what we discovered is this wasn't detrimental, but it actually removed amyloid from the brain. And so the panels on the right hand, the top left panel A shows you the amount of amyloid. All of that dark staining is amyloid present in the hippocampus of a transgenic mouse. And when we activated the immune system, the panel B shows you how much of the amyloid was removed by activating the activating the immune system. And the panels on the right are just a different way of looking at specific kinds of plaques or amyloid deposits. So we're really interested in, just activating or inhibiting the immune system is not going to be sufficient as a therapy for Alzheimer's disease. And if we're going to develop a disease-modifying treatment, we need to be much more selective in how we push the immune system in one way or the other. And so the inner circle here shows you different aspects of Alzheimer's disease that we are interested in. Certainly aging and amyloid and tau are the direct, amyloid and tau are the direct causes of Alzheimer's disease, but we're very focused on regulating innate immunity. It would be nice to have neuroprotective agents as well. And so in the circle on the outside, it sort of lists different strategies we have for how we can approach modifying these factors as therapeutic targets for Alzheimer's disease. And of course, one obvious way is with small molecule drugs. Another way that, well, go back, another way that we have done this very effectively for many years in the transgenic mouse models is using immunotherapy. And you've probably heard about how some of these immunotherapeutic approaches are being tested in clinical trials in human patients. We've also done quite a bit of work with lifestyle modifications. This could be changes to the diet or compositions of diet. We've done a lot of work in that area. And finally, when we don't have a small molecule drug and we want to modify a biochemical pathway, we can use gene therapy. And my lab is very active in developing viral vectors to deliver potential therapeutic strategies or to identify targets based on transferring genes. Okay, go ahead. So a typical experiment in my lab would be to treat experimental animals for a period of time with an experimental therapeutic to affect Alzheimer's, particularly innate immunity in Alzheimer's disease. We want to know if these therapeutics can cause a functional benefit to the animals. And so we do a lot of behavioral testing. We can test sensory motor function, motor capacity, a lot of, we have a lot of behavioral equipment in our behavior core in our vivarium. And one test that we developed is this radial arm water maze. Kind of what the maze looks like is on the upper left side, where it has 6 swim channels. We fill this up with water. In one position on the lower panel marked with a 2, in arm 2, there is an escape platform where animals can climb in onto this escape platform, get out of the water, and that is their reward for navigating this maze. They use spatial cues on the walls around the maze to learn how to do it. So we can start the animals in different arms. The diagram shows an animal starting in arm 4 and then swimming to arm 2 to attain the goal platform. And then the kind of data we generate is on the right-hand side, and the dark circles and bars, the groups really don't matter in this context, but the dark circles and bars are control animals. They make very few mistakes. They learn very quickly over a series of trials to find the platform more quickly with fewer errors. But a lot of our transgenic animals that express Alzheimer type pathology, whether it's amyloid or tauopathy, are impaired in this task, and they're slower to learn, and they actually never attain perfect performance by the end of our testing. Once we examine that, you can go to the next slide. We do a variety of measurements of things. We're doing a lot of work in my lab with real-time PCR and digital droplet PCR, targeted microarrays to identify gene expression changes that occur. We're dabbling with fluorescence in situ hybridization, and we do a lot of immunostaining and silver stainings and quantify that. So the outcome would be, does our experimental therapeutic affect the amount of pathology or the gene expression that results from that? But I would really like to go into the single cell RNA-seq. And so I've just purchased a benchtop cell sorter, and my lab is busy separating microglia and astrocytes from the brains of various experimental conditions. This is not our setup, but this is the setup that we have that's similar to it that I poached off the internet. But so we're definitely moving in the direction of trying to do some single cell RNA-seq work. I didn't put a, I think I'm done. I didn't put a, you know, all the people in my group slide, but I work very closely with Dave Morgan and also the other members of the department. And we have a very vibrant and active group of researchers in my group. So that's, I'll finish there.
That's Scott.
Yeah, hi everyone. Thanks for the invitation. I'm Scott. Broadly speaking, we study causes and consequences and potential interventions for addressing subcortical projection system vulnerability in MCI and AD. The two main systems we study are the cholinergic basal forebrain and the noradrenergic locus coeruleus. Both of these nuclei project to the cortex and hippocampus and amygdala, where they transmit, acetylcholine or norepinephrine respectively, to mediate memory and attention and arousal and vigilance. And of course, they degenerate early in the disease process. And as you can see in the bottom panels, they are exquisitely prone to tangle formation. So on the left there, you see in the blue-black profiles, those are sort of the typical globose tangles that you see in the basal forebrain. And then over on the right, on the bottom panel, those are the tangles in the blue-black profiles with the arrows, the more sort of fusiform or multipolar cells in the locus coeruleus. And there's a lot of data to suggest that the coeruleus is the very first site of tangle pathology in progression of AD. And so in the interest of time, I'm just going to concentrate on the latter system and actually just look at one aspect of that as just sort of an example of what we do. So in addition to looking for mechanisms of selective vulnerability, we also look at consequences, right? And so one of the things we're interested in are the cerebrovascular consequences of coeruleus degeneration in MCI and AD. And so just a brief introduction. So, locus coeruleus is Latin for blue spot. It sits right there in the dorsal pons, and it's a very tiny nucleus. It stretches from just the inferior colliculus to the velum along the wall of the fourth ventricle there. And there are only about 35 to 40,000 of these neurons, and yet they provide the major source of norepinephrine to the entire forebrain, as I mentioned, through these diffuse and heavily bifurcated axons, right? And so next slide, please. So we already know that locus coeruleus loss is prominent by the time a person comes to autopsy with mild cognitive impairment. In the case of our particular study, and others have shown this as well, we had amnestic MCI subjects, amnestic being the adjective for amnesia, so it's a memory loss more referable to Alzheimer's disease. And we show by a stereology that there was about a 35 to 40% loss of these neurons as you transition from no cognitive impairment, NCI, to amnestic MCI. And I can tell you with not going into too much data, but what we found was that the decrease in coeruleus neuron number correlated strongly with antemortem performance on neuropsychological test scores. In fact, our regression modeling showed that it predicted the extent of performance on not only global cognition, but also certain cognitive domains such as episodic and working memory. Next slide, please. And so why do we have this ridiculous hypothesis that coeruleus degeneration would impact cerebrovascular function? Well, there's actually a pretty good rationale, and it stems back decades. So we know through different methodological modalities that the locus coeruleus provides central regulation of blood flow and permeability, blood-brain barrier maintenance, and neurovascular coupling. And on the right here, what it's showing is, this is from our lab, this is the rat prefrontal cortex, and this is showing dopamine beta hydroxylase, or DBH, in red, and that's the rate-limiting enzyme for norepinephrine synthesis, so it's a good marker for those axons. And then we have a blood-brain barrier marker in green, which is SMI71, and you can sort of see those noradrenergic axons just wrapping around these vessels, right? And so, you have about 300 miles of, or over 300 miles of just capillaries alone, not even thinking about arterioles and everything else. And so, so in other words, if you took all the capillaries in your brain out and stretched them end on end, you could literally get from Boston down I-95 to Philadelphia. So, that's pretty amazing to think about. And it also begs the question, what could possibly go wrong there, right? And so we think that one of the consequences of Serratus degeneration is this impact on cerebrovascular function. Next slide. So to model this, we used the TG34419 Alzheimer's rats, and these guys express the APP Swedish mutation and the PS1 delta 9 mutation. So they have the lots of plaque pathology and behavioral impairments and things like that. And so without going into the lesion paradigm too much, what we did is we selectively targeted the locus coeruleus by using this aminotoxin, which is an antibody to DBH, the dopamine beta hydroxylase, and it's coupled to saporin, which is a ribosome inactivating protein. Some pretty legacy technology, actually, but we're just repurposing it for some more exciting studies, I think. And so what you can see is on the left, that's our control antibody conjugated in panel A. And you can appreciate the preservation of the noradrenergic axons in that inset there. And then as compared to the loss of the axons in panel B in that inset. And then when you go down to C and D, you can see the loss of the rostral forebrain projecting Serratus neurons, which replicates what you see in the human condition in Alzheimer's disease, compared to the preservation of the Serratus in panel C with the control lesion. And then in E, you can see where quantitatively, you lose that DBH signal in the cortex where the injection site is. And I should mention that the injection site here is in the prefrontal cortex, which is a major reciprocal connecting region for the Serratus. Next slide. And so what we found was that this lesion paradigm induced multiple markers of forebrain vascular pathology, as well as cognitive impairment in these quote unquote Alzheimer's rats, right? So in the top left here, those little black gumballs, those are the amyloid plaques, but then you can also appreciate with the arrows, the vessels that also have amyloid staining in those. And so that represents the emergence of cerebral amyloid angiopathy in the lesioned animals compared to the controls. And that's sort of shown there in the quantitative panel on the right. Yep, that's right, yep. And then moving down to the next figure, down, thanks, Irving. We also showed that the lesions induce markers for blood-brain barrier leakage. So the green marker is our blood-brain barrier, the red is our albumin, and so B is the lesioned animal. And so you can see this blood protein out in the parenchyma of these, in the brains of these animals compared to the lack of that leakage in our control animals. So that was pretty exciting. And then finally, in the bottom panel there, Irving, what we showed was that the lesion resulted in an increase in the wall-to-lumen ratio of parenchymal arterioles. And this is a commonly used marker in cardiovascular research to show vascular remodeling in the presence of cardiovascular disease, as well as stenosis or a lack of vasoreactivity, arterial stiffness, right? And then all this led to impairments on the Barnes maze in these animals, or they were related to the Barnes maze impairments. So we saw in panel B there, the lesioned animals, it took them longer to find the target hole in the spatial memory task. And then in panel C there, they spent more time visiting holes they'd already visited, looking for the target, which is considered a working or procedural memory task for these animals. And so our regression modeling for this showed that all of these vascular markers, the extent of the pathology, predicted the extent of cognitive impairment as shown on the Barnes maze in these animals. And so some new and ongoing directions are to look at some sort of standard intervention studies like norepinephrine replacement. We're doing physiology of parenchymal arterioles with Dr. Doritz over in East Lansing. And she's actually also showing that there is a lack of vasoreactivity in these vessels from lesioned animals. We're also doing in vivo perfusion and functional MRI, small animal imaging with Drs. Wiseman and Zhu over in East Lansing, as well as some other more mechanistic studies to try and drill down on what are some of those molecular pathways that are driving these effects. And so I wanted to kind of circle back to the cerebral amyloid angiopathy and show you a little teaser bit of data here. And so we think that this impact of Serralis degeneration on amyloid angiopathy may be due to an impact on its regulation of vascular drainage pathways. And so these are actually wild-type rats that received either the control or the Serralis lesion. And then a month later, we injected fluorescent A-beta 1 to 40 in the same surgical site. And so what we find, and just to give you the key there, so green is our smooth muscle cells, blue is our endothelial cells, and then red is the fluorescent A-beta. And what we're finding is if we move like 200 microns to the either posterior or anterior to the injection site, we see this, if you look on the right, there appears to be a retention of amyloid in the vascular wall. Okay, and so that suggests, again, and cerebral amyloid angiopathy is typically found in the basement membranes of smooth muscle cells. And in the control animal, we're really not seeing a lot of evidence for that. I showed this particular image because when we do see a signal for the amyloid near a vessel, it's usually kind of just sort of diffuse, and then over on the left panel, just sort of diffuse and not really incorporating, maybe a little bit there at the bottom left, but bottom right. But that's just sort of a teaser. Of course, we need to carry these out and quantify them more. And this is done with Rots Carrari at Southampton. Next slide. And so just to acknowledgement, you probably saw the name Kelly a bit on the slides. That's Sarah Kelly on the left over there in the picture. She actually just passed her bar for intellectual property law down in Washington, D.C. And then that's Aaron and John in the picture, and then some of our newer members, Masa and Batul. And then just want to thank our Alzheimer's group who are here today, and then some of our MSU collaborators and external collaborators and the folks at the Michigan ADRC, as well as our funding sources. So thank you.
Next, Dave. You're muted right now.
Of course. Thank you all for inviting me to talk to you today. My name is Dave Morgan. I'm a foundation professor here and also director of the Alzheimer's.
You're muted again for some reason. No.
I have a few disclosures. I'm an MPI on an SBIR grant to a human on a chip company. We get contract funding from a pharmaceutical company, and I've consulted with AbbVie and Biogen. Next slide. So you heard Dr. Gordon talk to you about immune activation in Alzheimer's disease. And in full disclosure, she and I have been working together for 35 years. We're married and we have children and all those things. So if there's some overlap, that's why. But this whole immune activation hypothesis has a fair amount of supporting evidence from neuropathology in the 1980s, the epidemiology finding reduced risk associated with people who need NSAIDs or TNF inhibitors. And then the GWAS study she mentioned showed very nicely that innate immune proteins can increase or decrease the risk of developing Alzheimer's dementia. And then finally, when we've done manipulations of the innate immune system in mouse models of amyloidosis or tauopathy, I hate to call these models of Alzheimer's disease because they're not. But those manipulations can affect the core pathologies that we find in Alzheimer's disease. Next slide, please. So one of the things that's very interesting about innate immune activation in Alzheimer's is we've got all of these wonderful descriptions of a sequence of events that have occurred in Alzheimer's disease. Yet there's nobody who's really drawn where innate immune activation is occurring here. And on the next slide, I think part of the problem is that the biomarker that you would use to essentially signal for innate immune activation would determine when it would show up on that slide. On the right-hand curves, we have a study we did a while ago looking at old mice that have amyloid in their brains using an anti-A-beta antibody treatment over 3 months. And during the time course of this, we have biomarkers for innate immune activation that go straight up. We have biomarkers that go straight down, and we have some that go up. And then as the amyloid is completely cleared from the brain, go back down. So it's hard to know exactly what you would use to characterize this. And I think we're increasingly recognizing there is tremendous complexity to these immune responses. Next slide. And oftentimes, this is a review article that we did with Dan Lee talking about immune regulation and amyloidosis. But we also mentioned here that very often the same manipulations that have an effect beneficially on amyloid can have a detrimental or exacerbate the tau pathology that we find in the tauopathy models. And we think that there are some aspects of the innate immune response to amyloid which mediate some of its influence on the development of tauopathy. Next slide. So one thing I'm very certain of, I can tell you that unless you're one of these two organisms pictured on this slide, chances are that evolution never had a chance to select for an optimal innate immune response to neurodegeneration. Severe head injury in most of us would have resulted in probably, at least during the times when we were evolving, probably a rapid death. And yet somehow these organisms are able to survive. Interestingly, woodpeckers do have tauopathy that looks like chronic traumatic tauopathy, and they don't seem to suffer from it cognitively. Not that I've tested cognition in woodpeckers, but we think that there are ways that we can protect ourselves from this, but that it's unlikely that this was ever selected for intentionally. And I talk about this a little bit in a review article that came out a few months ago in Alzheimer's and Dementia. Next slide. So certainly humans who have neurodegenerative factors in their brain, they do have responses from the innate immune system. We see them, but they're probably not coordinated in a specific fashion. The role of natural selection on innate immunity was to help during development, and that is to guide neurons to various places, to clear out the circuit of neurons and synapses that are produced, to leave only those that are properly connected to survive and recycle the products of those dead cells back into the living ones. And then in adulthood, it's designed to protect us from the constant assault by parasites and bacteria and viruses that until recently, almost every living human had chronic conditions that were associated with this to protect them from. And so the responses we have to neural injury, such as this gentleman here probably is suffering, are haphazard. They're just simply responding to whatever signals happen to be present, and they're probably not activating a coordinated, genetically controlled program, but instead they're having, some things that go up, some things go down, and none of them were actually intentionally developed for that purpose, which is why often people say, well, it's Janus-based inflammation. But there is no real repair program that evolution ever guided. Next slide. So we probably have some of these things which are beneficial and some of these things which are detrimental. This is some biomarker work that I've been collaborating with a group at McGill University on. And in the very left-hand slide on the Prevent AD slide, on the y-axis is people who have no amyloid and no tau. And then stage 1 is where they have amyloid only. And then stage 2 is where they have amyloid and tau. And surprisingly, the people who developed amyloidosis were the ones that had the lowest levels of these markers. Now, this could have been induced, or it could have been a trait that they developed. Nonetheless, as the tauopathy developed and neurodegeneration developed, these markers increased. In panel B, you can see similar results from the ADNI data set, in which, again, the transition from normal to amyloid only seems to be associated with lower levels of many of these innate immune markers. And the ones that are solid lines actually are thought to be suppressors of innate immune activation. And they went up and then went down. There's a whole series of references that you can read in the review article that I wrote that talk about other markers that show a similar pattern. Next slide. So one of the key elements that Pierre Meyer and John Breitner and Jude Poirier did at McGill was to try to identify an index of these immune markers that might predict cognitive decline. In amyloid negative people, cognitive decline varied across individuals to some extent. It was relatively small. but the index didn't predict it. But in the amyloid positive population, what they found was that individuals that had higher levels of these immune activation markers had the slowest rates of cognitive decline subsequently. The same was observed in the study by Hamlin et al. Using the TSPO ligand that measures presumably microglial activation, although there's some question in my mind about that. Nonetheless, they divided their group into a fast declining group and a slow declining group, and they found that the slow decliners had more TSPO, reflecting greater activation of the innate immune system. Next slide. So the AMP-AD program has been carefully documenting in an unbiased fashion RNA and protein and lipids and all kinds of things in Alzheimer's disease brain sets. And they've come up with about 400 possible drug targets. Now, somehow we need to winnow this down into something a little more likely to result in a positive outcome. Genetics is one way to do that. But another way to do that is to try and search for those markers that are associated with rates of cognitive decline. And increasingly, studies are coming out with CSF samples, blood samples, and postmortem tissue that have found a number of markers in large populations of 300 or 400 people that seem to be associated with the rate of cognitive decline. Either high levels predict more rapid decline or slower decline. We do know that the AD core pathologies, amyloid and tau, do predict the rate of decline because they're also predicting the rate of neurodegeneration. And so these other proteins have two possible outcomes. One is that they're essentially state variables, that they're responses to the ongoing neurodegeneration, and that they're essentially telling you that the people who have higher rates of neurodegeneration decline more rapidly. But the other one is that they might actually moderate the vulnerability to the AD core pathologies. Much like we might find in those woodpeckers that I showed you before. And that these are traits that people have that have different levels of these proteins naturally. And this is conferring some type of resilience to them. And so what we're doing in our system right now is we're trying to distinguish which of these proteins associated with rates of cognitive decline are state proteins and which are trait proteins. And we're doing this using multiple model systems, amyloid models, tau models, and normal aging cognitive decline models. And we're using manipulations that are more in line with what a pharmaceutical agent might be able to do. We're not doing global body-wide knockouts of proteins to try and figure out what they're doing. We're doing very cell-selective knockdowns and doing it at times when they're adults rather than doing it when they're still developing. And similarly, we're upregulating by having only partial but brain-wide increases with an AAV called PHPEB. And we hope with these multiple models that we can see which of these manipulations really are having effects on amyloidosis, on tauopathy, or in some cases, we may find they don't affect amyloid or tau, but they still protect them from cognitive decline. And these are the types of things that we're looking for in our research group right now. And I thank you by showing you a slide that actually includes all the people that Dr. Gordon forgot to include in hers. And so this is our joint lab on the left-hand side, and then some of our collaborators on the right and our supporters. And I thank you very much, and I apologize, but I have to leave because I'm 5 minutes late for another meeting. So if you have questions, you can email me at scientist.dave@gmail.com. Thank you very much.
Thank you, everybody. That was great. I think we have a few minutes for questions. I want to ask one question before we answer to the two questions. Maybe, Irving, you can take a look at the QA and see if you can address those questions. So the question that I have is actually for Marcia. So maybe wrap it up. So I was kind of like wondering, so excellent presentation. I think that the research and the science is amazing. Thank you so much for taking the time again to give the presentations. I'm very interested in general in everything that has to do with the diverse populations and hearing everything about immune markers. it made me think about like, I don't know, like the association with immune response and like chronic stress and how sometimes the minoritized groups are more exposed to chronic stress. And I was wondering if you have considered or had a look into the different response in different diverse groups into immune response, or are considering that for the predictions or cognitive decline, or what David was talking about, like, you know, how to predict rates of cognitive decline over time in different groups.
As far as I know, there is a very strong literature showing effects of stress on the adaptive immune system, but I don't know of a whole lot of research that has looked at the innate immune system and its responses to stress. So that's a very interesting question, actually, that I think we know very little about.
It will be really cool. I don't know if this is like this is not my area of research, but it will be really interesting.
Yeah, absolutely.
Thank you. Irving, you want to try to go
Yeah,
over
there
the
was
questions?
there was one question that says, what opportunity postdocs have to work with, be involved in the training of PhDs or undergraduate students? So our postdocs, like graduate students, go into a individual development plan. And when we identify that one of the career goals is to go to academia, we make sure that that's part of the training process, to be involved in the training and even to have teaching experiences at the medical school. We do have undergrad students that are not from MSU because we are in Grand Rapids, not in East Lansing, but there is undergraduate institutions here in town, Grand Valley State University, for example, that there are undergraduate students that actually come to our labs, especially in the summer, to do internships. And they usually work with senior graduate students or postdocs. But it's based on what the postdoc identify in the IDP. And we develop a committee that the postdoc have attached to independently or in addition to the mentor or the person that the postdoc is working, that we actually tailor the training based on the career development and make sure career goals, and make sure that we coordinate and provide the resources and the opportunities needed to develop those skills to move in that career path that the postdoc decide. Marcia, you want to add something?
Yes, I want to highlight also that within each lab and across our Alzheimer group within the department, we're really interactive and team-based. And so the model system where one researcher is off in a separate room doing their own project without any interaction is not the way that any of us operate here. And so there will always be opportunities for interaction and working together on projects across all levels. And I would say too that I also am the faculty liaison for a high school program. So not only undergraduates, but we have high school students come in the summer for summer internships that to have a research experience in the lab too. And so there are plenty of opportunities for interaction, team-based science, and communicating your knowledge to people coming into the field behind you.
Yeah, there was a related question about training opportunity for international students. And actually in my lab, I have international students. I know Scott and Nick also have international students, and Marcia had international students too. So there is opportunity for international students to come to MSU and study with us. Currently, we do have international students, and we support them in every way possible. There is another question from Hannah Stoker that actually she said that she's originally from Michigan, so she's a Michigander, and she's currently a postdoc at Heidelberg, Germany. And the question is for you, Marcia. She said, I was wondering if you have any experience with plasma GFP levels and if there is any relation to the innate immune response. Marcia?
I do not have any direct experience, but we look at brain levels of GFAP all the time. Yes, I'm very interested in that. Yes, very interested, but I don't have anybody working in that area right now.
And I'll add to that, that GFAP and some markers of inflammation are going to be part of what the biomarker core does for the Michigan Alzheimer's Disease Center. So we will have data at some point in the future on some of those kind of markers.
Plasma and CSF and the two?
Plasma. Yeah. The center is mostly focused on plasma.
I mean, in the big picture, I think we would like to do CSF work too. And we're working on generating the interactions and the team-based sample collection that would be useful for such a thing. So yeah, I mean, we're interested in that. It's just our state of Michigan is more focused on blood at the present time, but we're definitely interested in moving in that direction.
Perfect. And because of time, we had to close. Scott, I have a question about the locus coeruleus, but I think that I'm gonna send you an email about it. We are actually doing a study with the locus coeruleus in the Colombian kindred, and I will be very interested in hearing your thoughts about.
Yeah, no, Heidi Jacobs and Keith John, those guys just put out a really awesome paper on that.
Yes.
So yeah,
we are doing something similar, but they're taking it also to the cerebrovascular function. So definitely very interested in following up with you in that paper. But hey, thank you everybody for joining us again today. I think that we had to close now. Irving, thank you for putting together an excellent webinar. And thank you, Dr. Gordon.
And yes, I don't know if it's highlighted, but you can always find our departmental website and at msu.edu and look for our Department of Translational Neuroscience. And then any contact information will be listed there. So feel free to contact us.
Thank you.
Thank you, Jackie.
Thank you.
Have a good day.
Take care.
Bye-bye.